Hello student, in last lecture of taxonomic procedure part first we see the how animals are collected. So in today's topic we talking about the how animals or space different kind of species preserved in the museum for their identification and study of biology. So basically these species are useful for the study of biology by their morphology, anatomy, embryological evidence, genetics. So basically these animals preserve long time duration for the study of human being. So let's start today's topic, that is taxonomic procedure part second. So in today's topic we're talking about the types of collection and how animals are preserved in the museums. So most biological collection are either dry collection or wet collection. They also may include collection preserved at low temperatures or microscopic collection. So basically all the collection of species are essentially preserved in the museum for avoid the degradation or decomposition of in that species. So basically these species are preserved in the museum for the better study of human being. So basically these types of preservation containing the four types of preservation, that is dry collection, wet collection, biological low temperature collection, and fourth is a biological microscopy collection. So first of all, that is dry collection, how the dry specimens are preserved in the dry state. So basically dry collection consists those specimens that are preserved in a dry state. So basically dry collection containing the specimen or species that are preserved in a dry state without having the water content in their body. So basically two factors influence the decision about the preserving specimen this way. So basically two types of characters that have to influence the we have to think about the how animal have to preserve. It may be dry or wet. So basically first of all, that is rigidity. So rigidity is important part of the preservation of animal. So rigidity, so some specimen can be preserved naturally, for example starfish, or artificially with the sufficient rigidity to accommodate normal handling. So such a specimen often are suitable for the dry preservation. So basically first part, that is rigidity. Rigidity is important for the specimen preservation. This rigidity provide the strength to the species for long time duration. This rigidity accommodate for the normal handling of such a specimen for the human being also. So such a species are used mostly suitable for the dry preservation, and for example that is starfish. So starfish is naturally preserved by its own because this sea containing the salt, and salt preserve those animal after their deaths. So this organism containing the rigidity in their structure, and they get easily preserved for the dry collections. Second type, that is specific characteristics. So during may provide the best available means to preserve the natural color. So dry collection is essential for the preservation of natural colors of the animal or species, because during wet collection these natural color may be disappear due to the chemicals we use for the preservation. But a dry collection provides such a character for the best available for the preservation of natural color, and also the distinguishing features, that is skeletal parts or surface details, because these skeletal parts of surface details can be get vanished due to the chemicals, and such as specimens in a dry state may have the great potential for the interpretation and the research. So dry collection is type of best character for the such animals which containing the natural colors of variation, that is butterfly. So butterfly is a best example because we know that the butterfly is having the different types of natural color, and for study or interpretation and research of for such a butterfly by the human being, we have to maintain their natural color for their identification and classification, because their classification is depend on their body color, body structure, and their natural colors. So basically, dry collection is type of collection for the preservation of such animals. So this is the figure of dry collection in a museum, which is useful for the animals preserved in the dry state for the study of further study or research and interpretation by the human being. Next part or second type, that is wet collection. So wet collection is bullied preservation, which specimen kept in a liquid preservative to prevent their deterioration. So basically, wet collection is a type of collection in which we kept the specimens in liquid preservative such as a formalin. Okay, mostly the formalin is used for the preservation of such animals or species in museums. So basically, wet collection is a type of collection in which we use the different types of chemicals for the preservation of animals. Mostly, certain biological specimens are preserved in a wet form due to the convenience, because sometimes these biological specimens may be decomposed due to their presence of bacteria in their body. So we have to preserve in a wet form due to the convenience study for their anatomy and morphology. So we have to preserve them in a wet collection or in chemicals for the to avoid the deterioration of such animals. So in intent to preserve the body from and soft part for a variety of use, when the color preservation is not critical or dry preservation sacrifice quality needed for other intended use, fluid preservation is beneficial. So basically, dry collection is kind of useful for preservation of natural color, but wet collection is more useful for the preservation of body form and soft part of variety of body organs in the body of animal. So basically, this wet collection is useful for the preservation of body form and soft part, because due to the dry collection, the water get lost from the body of organism, and due to that the cells get shrink, and so that the organs or soft organs cannot be s- further studied by the human being. So wet collection is kind of useful for the study the soft parts of the animals, because we can sacrifice the some quality of that cannot be more useful for the research purpose. But this, and whenever we study the soft part of the animal, we have to use the wet preservation is kind of useful, because chemicals preserve the salt part as their own state. Okay, so that we use the wet collection more in the museums for the preservation of such a species. So this is the kind of wet collection. So in this figure you can see that these bottles containing the chemicals such as a formalin, and in these bottles the organism are preserved. Next part or next type, that is biological low temperature collection. We know that at low temperature body cannot degrade or decompose. So low temperature maintain the body structure of specimen. So specimens are maintained at the low temperature to preserve is useful for the preservation of animals. So low temperature is useful for the maintain the specimen in its entire structure. So basically, the soft parts for various biochemical analysis is useful for the research and interpretation of anatomy of organism. So basically, the for the study of soft parts of various biochemical analysis, mostly the museums use the low temperature biological low temperature preservation method. So in low temperature preservation method, whole organism can be live in its state, that is it able to live and grow in that low temperature collection. So basically specimens preserve at low temperature. For example, some algae, some protozoa, that is specifically parasitic strains, viruses, clone viral genome, bacteria, bacteriophage, plasmids. So these type of microscopic animals are preserved at the low temperature, and they can be grow by are able to live and grow in their viable state from the low temperature preservation. So basically, also in low temperature collection we also preserve the animal tissue, which is from the dissected organs or muscle cell lines. So these are also preserved in the low temperature. Also the blood and blood components, because blood and blood components can be degraded in the room temperature, so that we have to maintain the low temperature for the preservation of blood and blood components such as whole blood or serum, plasma, antiserum. Also we preserve the semen and venom for the preservation of their biological activity. Also, the other samples such as the clone probes, isolated proteins, nucleic acid, cell suspension. So these all these substances or all these material are saved or preserved at the low temperature for maintain their biological activity. So basically biological low temperature preservation method is useful for the different kind of processes occur in the lab. Next, that is last one, that is biological microscopic collection. So basically microscopic animals are preserved on the glass side, that is microscopic collection, and we use this biological microscopic collection or preservation method for the identification of such a parasite, such a microorganism, and their further studies. So basically scientists preserve certain specimens as a microscope preparation to preserve the whole and partial organism. So basically scientists preserve such a certain specimens like plasmodium, eggs of insects, or other microscopic animals for the study of micros in the study in under the study of microscopy. So basically this preserve the whole and partial organs in during microscopic collection. So basically various kind of microscopic examination done by the microscopic collection preservation method. So some kind of biochemical analysis including extraction of DNA is also done by the preservation of organism in microscopic collection. So specimens prepared for the microscopy may found in all biological collection but are most common in this collection. So entomology, mycology, parasitology. So these study of the entomology, mycology or parasitology is essential or based on the microscopy. Basically we study the slide or microscope and this slide and the insects on the slide. So all this study of this entomology, mycology and parasitology are depend on the microscopic collection or microscopic preservation, so that scientists preserve the such a specimen and microscopic animals in the microscopic collection or microscopic preservation form. So biological microscopic collection, it is also commonly called as microscopic collection, to be auxiliary to more traditional collection. So basically the traditional collection that is microscopic, and we usually use this kind of preservation of animals or organism, that is microscopy preservation. For example, such a auxiliary collection includes the histology, karyology scales. So during histology and karyology, karyology means nucleic acid study. So for the microscopic collection, we in this study of the histology and karyology or scale of tissue, we use the microscopic collection preservation method. So these methods are usually used for the study of histology, karyology and scale. So in this figure you can see that these are the slides you usually prepared for the study of parasitology, entomology, histology, karyology, and these slides are more useful than the theory knowledge, because we On the study of microscopy, we see the entire organism under the microscope and easily identify them by their body structure. So these animals are preserved on such a slides for long duration. So that is for today. So in today's lecture, we're talking about the preservation methods of taxonomy. So taxonomy procedure containing the four types of preservation method, that is dry collection, wet collection, low temperature collection preservation, and last one, that is microscopic preservation. So these preservation methods are more useful for the preservation of different kinds of specimen in the museums. So that is for today. Thank you.